Hello! It's me, Daniel,
And me, Jonathan.
and we're going to present you a project of probability and genetics using sheets and coins.
Hello!
We're going to present you two projects.
One about flipping two coins at 20 times and flipping two coins 100 times to see the probability to get heads heads, heads tails, or tails tails.
Hello guys, so this time we're going to use 200 sheets, sheets, 100 red and another 100 white. And we're going to put in a container 50 red and 50 white, and in another container 50 red and 50 white. The deal with this project is to take off one sheet. It needs from it container, and if it gets, for example, red and red, the red is dominant, so it's going to be an homozygous red. And if it gets red white, it's going to be an heterozygous red white. And if it's white white, it's going to be a homozygous recessive white. Yeah.
Why are your real results for the coin flipping and the genetic experiment different from the predicted results?
Well, for science, everything is joined to count 100. So in this case, we're going to divide 100 between 4 categories: heads head, tails head, head tail, and tail tail. But because head tail and tail head are actually the same thing, we're going to just add them, and that will get 50% out of 100. So they will differ because we won't get always the same answer. We will never get. We will never have. If we throw a dice, we will never have 6 all the time. It will differ with 1, with 4, with 5, maybe. Just probabilities. What difference in result would you expect if you use 100 items of each color in each can instead of only 50? Explain your answer.
The results will be the same as with the 100 sheets, because the probability stays the same, because it is only 2 times less the amount of sheets. So the results can vary a little bit, but the probability will still be the same. Achilles, for your last question,
Yeah.
assume the red and white beans represent alleles for the codominant trait. This means RR will always produce pink flowers. What is the predicted result in genotype and in phenotype? And what results did you get?
For the last experiment of the sheets, we used 200 chips, as we said before. 100 were red and another 100 were white. As a result, we get 26 pairs of red dominant, yes, homozygous. For the other result, we get 28 pairs of white recessive, and for the last result, we get 46 pairs of heterozygous red-white. That was 50%. The result for the allele will be 46 pink flowers